నాకు రివర్స్ కనపడుతున్నాయండి. నాకు అర్థం డోంట్ సే ఎనీథింగ్. షేర్ స్కీమ్ అన్నప్పుడు ఏమది ఆయనది ఎందుకు మ్యూట్ చేయాలి? అది మన ప్రాపర్టీస్ కాదు. ఓన్లీ 6 participants are there. You bet. No. Speaker view. Speaker, not gallery. Speaker. And also you put it full screen. Full screen on చూడు అక్కడ.
సార్ మీరు ఏ నెంబర్ తో జాయిన్ అయ్యారు సార్?
మీరు పంపించారు కదా 606.
అది కాదు సార్, ఇక్కడ మీ ఫోన్ నెంబర్ సార్ ఇప్పుడు మీకు కోవోస్ట్ ఇవ్వడానికి.
నా ఫోన్ నంబరా 98491.
అది కాదు సార్, ఆలివ్ జెసికానా సార్ మీది? జాయిన్ అయ్యింది ఇప్పుడు?
ఆలివ్ జెసికానా ఇది? ఆలివ్ జెసికా, ఎస్.
ఓకే
5A5. ఇది ఒకటి ఉందా మళ్ళీ? ఎందుకు ఇప్పుడు షేర్ స్క్రీన్ చేయాలి కదా మనం, అది రావు. Should I share the screen?
Sir, 1 minute. Okay, very good afternoon all my teachers. A warm welcome to today's session for the afternoon session. And Professor Siddhi Veeresham sir, the principal, SRM University, Chennai, previously worked as a principal and dean in the Kakatiya University, Warangal. With these words, I am welcoming the Professor Veeresham sir and Mrs. Amini. Introduce the guest and today speaker for the faculty members. Sir, screen reverse లో ఉంది సార్.
అది ఏందో నాకు అర్థం కావట్లేదు. రివర్స్ లో కనిపిస్తుంది ఎందుకు? Why it is in reverse? I don't know.
సార్ మీరు చెక్ చేయండి సార్ అక్కడ.
నా దగ్గర ఏం చేయాలి? ఏముంది చెప్పు. స్టార్ట్ వీడియో. I don't know, it is. I don't know. Anyway, should I share the screen so that you will be able to see? Gopinath? Slides so that we know what it is. Here is slides. Ide kada. Okay, this is fine. Video on time. Are you able to see my screen, Gopinath? Gopinath?
Yes, sir. Screen is visible.
Okay.
Okay, then, sir, please wait 1 minute.
It's okay. I will introduce myself.
No. Okay, sir. No.
It's okay. Fine.
In the session is the part of biography also there, sir.
No problem. That is not a big deal. Big deal.
Okay. Pranati. Sir, Pranati, proceed and.
Good afternoon, sir. Okay, sir.
Speak loudly.
Good afternoon, Veeresham sir.
Oh, very good afternoon, Pranati.
Myself, Pranati sir, member of faculty, Jntua, Otpri.
Okay.
Thanks very much, everyone. It is an honor to me to introduce our next guest and speaker, Professor Siddhi Viresham Garu. He is a principal and professor in Sri Ramachandra College of Pharmacy, Institute of Higher Education and Research, Porur, Chennai. Dr. Sri Viresham Garu was born on 8 August 1959 in Kannaram, Karimnagar. He has obtained B.Pharmacy degree from Kakatiya University in 1984, M.Pharmacy from Birla Institute of Technology, Ranchi in 1991. Viresham sir got PhD award in pharmaceutical chemistry in 1991 from Kakatiya University, Warangal. And also sir got 2 postdoctoral fellowships from USA Cornell University in 1994 and 1998. He worked as an assistant professor in Gurbaga University, Karnataka and Kakatiya University, Warangal. Also, sir worked in various universities as associate professor and professor. He has been life member of Indian Pharmaceutical Association, Association of Pharmacy Teacher of India, Indian Society for Pharmacognosy, Chromatographic Society of India, Indian Associate for the Study of Traditional Medicine, Telangana Academy of Sciences, and also he is member of Editorial Board, Indian Journal of Natural Products, Secretary of Indian Pharmaceutical Association, Warangal Branch, Executive Member, Pharma Alumni Association and Vice President, and member of International Association for the Advancement of HPTLC. Viresham sir has 27 years of research experience in the field of pharmacy, 32 years of teaching experience, expert member in AICTE for the evaluation of major research products. Sir, chairperson board of studies in pharmacy, faculty of pharmaceutical sciences, Palamuru University, Mahabub Nagar. Sir delivered a number of lectures on national and international seminar conferences, workshops across the country. In 2014, he got Manjushri Pal Memorial Award for the Best Pharmaceutical Scientist from APTA and SPAR Eminent Teacher Award. AP Coast Pharmaceutical Scientist Award 2011 from Government of Andhra Pradesh. Best Research Paper Award 1992 from IDMA Mumbai. Best Teacher Award in 2015 by Government of Telangana. Sir has 146 publications. Sir presented 66 presentations in various national and international conferences. Sir published a book on medicinal plant biotechnology in 2004 and one more on phytopharmaceutical for diabetic complications in 2020. 25 PhD students were completed their research under the guidance of Professor C. Viresham sir, and 5 PhD students are currently working. 80 master's degree students completed dissertations. Sir visited foreign countries, Malaysia, USA, Ethiopia, Japan, and Jordan on academic assignments. I, on behalf of an entire administration, faculty, and students, wholeheartedly welcome you, Viresham sir, to faculty development programs. Thank you, sir.
Thank you very much, Pranati, for introducing me to the audience. Thank you.
Thank you, sir.
First of all, I would like to thank my own friend, Dr. Gopinath, for inviting me for this FDP program of Jntua Anantapur. I welcome all the delegates, my teaching friends. As for the advice given by Dr. Gopinath, I just want to make you that he was trying to ask me how best I gone into my professional career right from my studenthood. Basically, if you ask me this question very openly, I'm going to tell you that when I was in 10+2, I wanted to become a medical doctor, which I could not become. Then I started looking for what is the other professional careers available at that time. 1 year I waited for after 10+2, and then I entered into B.Pharm in 1979 in Kakatiya University, Warangal, where I tried to finish my B.Pharm. And while I am studying my B.Pharm, I was really fascinated by the subject as well as my teachers who are very stalwarts like Professor D.R. Krishna and Professor Kokate, Professor Rambhav and Professor MCP and all. So by that, I inspired a lot to go for masters. I was doing my masters at Birla Institute of Technology, Ranchi, and many of my teacher friends agrees me that it was very difficult those days to get the master's program in pharmaceutical sciences. However, I have been got a seat in Birla Institute of Technology, Ranchi. Throughout the country, there were only 22 colleges offering the M.Pharm master's program that time. While I was in final year of my master's program, I thought of going into the PhD, and then I approached with Professor Kokate, and I started working on the research. While I was doing my research, I thought that the PhD degree which I'm working with full time could not be best for competing the scientific world. Then I thought that I should do the postdoc. So that's how I entered into the postdoc in Cornell University, Ithaca, New York. And then I'm really lucky to tell you that I have been entered into the top 10 university called as Cornell University, Ithaca, New York. And the advisor of mine is a very well-known professor called as Professor Mike Louis Schuler. And then my next postdoc was on the UNESCO back fellowship. And once I have done my postdoc, I was really competent to get the grants. And that's how my first grant of the research project was got into 94. Until date, whatever the projects of which I apply, I'm sure of getting into the funding. And I'm very happy to share you that even in Ramachandra, I was successful in getting a couple of projects during last 1.5 years. One of the project is DST FIST program. And this program is the first time in the history of Sri Ramachandra Institute of Higher Education and Research, as well as it is the 5th university in India, private university, to get this funding. And this funding is basically for 5 years, and afterwards it's again goes on into the different cycles. So that's how I try to publish a couple of books and a patent, as well as 25 PhDs and so on. Its only thing is I advise you a few things, like you should be very hardworking and dedicated, and you should be ambitious, and you should have a vision what you want to make. And if all the teachers are motivated, probably we can do the wonderful things. So this is what I exactly I asked to deliver, as well as the funding opportunities are many. Like DST is there, SACRB is there, UGC is there, AST is there, and so there are ICMR, CSR, almost all the major grants have been granted me by the various organizations of central government. And now that there are a lot of opportunities even today, if you can write a good proposal on your research area, and the project should have a novelty, and you should clearly able to explain your hypothesis, then it is easy to get the funding. So the funding mantra is you should have what are your publications into that particular area, and your publications should speak about you, then the funding chances will become very, very easy. And I advise all the faculty. What's up? All the faculty need to go ahead of applying the funding with a small funding, like 10 lakhs, 20 lakhs, then go on increase so that you will get the funding. So with this, I start today's my talk. And I belong to Sri Ramachandra Institute of Higher Education Research, where I was working as principal faculty of pharmacy. We have a fantastic medical center. It has about 14 faculties. And this is the university which is NAC third cycle, also A++ grade. And we have, this is a sports science. The sports science is the unique of Ramachandra. And this is what hospital, the aerial view of the campus, which is very beautiful. It's not going. Okay. This is the college where I belong, and then this college has been started in 1993, and it has about 49 faculty, and this is now called as Sri Ramachandra Faculty of Pharmacy. This is what exactly where I represent currently, but I'm also happy to share you that I have resigned for this particular position also in a couple of days back. And since this program has been fixed very long back, I am just trying to give you this. And this has the college has a lot of these things like NAC accreditation, MBA accreditation, AICTE, PCI, and all those are there. Even NRF ranking is 20th position. Now, coming to the topic, plants as a source of drugs and role of pharmacognosy. And I'm just trying to give you exactly the role of pharmacognosy. And many of our friends feels that pharmacognosy nobody will like to study. So I just want to make them to understand that probably pharmacognosy is the one which is the mother of pharmacy, and the all others are subsidiary branches of pharmacognosy. Now, what is the global, and many of the pharmacy postgraduates as well as the bachelor degree holders will try to jump into the teaching rather than to become entrepreneur skills. And there is a lot of entrepreneur skills are available if you can understand pharmacognosy very well. So that is the reason why I just want to give the global business rank where exactly you can place a role. Now, medicinal aromatic plants, global opportunity is there. See, for example, I have given all these areas. You can you can take a small area and you can try to do, for example, traditional medicine, complementary alternative medicine, household remedies, and herbal drugs as such, herbal drug preparation, and phytopharmaceuticals, modern drugs, drug leads, drug intermediates, and insect repellents, insecticides, pesticides, fungicides, and pharmaceutical aids like gums, mucilages, edible dyes, sweeteners, essential oils, flavors, fragrances, perfumes, cosmetics, and as such, if you can't do, it is extraction. Extracts also can be sold out into the market. So the business angle, if you look at anywhere else, you can, anywhere, wherever you want to feel, you can do your role. For example, if you see the world market for the medicinal plant-based drugs, phytopharmaceuticals, they are the natural products, wellspring of drugs and as well as drug leads. Now, if you currently see the drug leads and drugs, both are derived from the phytopharmaceuticals. Phytopharmaceuticals again derived from the plant source. There are a number of drug molecules which are developed during the 2006. If you try to see the data, 166 molecules have been originated, and this data might have gone into the double figure. Now, plant steroids will have something like 22 billion global market data. Now, one of the drug lead like paclitaxel, which itself will, paclitaxel is having about a $1 billion market. Vinblastine and vincristine amount around 100 million per annum. Nicotine and scopolamine patches have also combined sales is more than $1 billion. Now, many of our, I've feeded Gopinath as a Guntur Gopinath. Guntur is very well known for tobacco, right, Gopinath? I'm sure you understand that. So we can do something wherever you have that advantage. And if you see the available anti-cancer drugs, 40% of them are natural products, or 8% of them are natural product mimics. So this is the what really you should understand that. Now, if you try to see some examples of recently new plant-derived drugs in the global market, like artemether, which is a anti-malarial drug, galanthamine, which is obtained from Galanthus vernuki, and it's Alzheimer's disease. Nitizone is a modification of mesotrione, is an herbicide. Tritropium is a modification of atropine, and now it is used for COPD. Now, solamigrine, which is obtained from Solanum species like Solanum xanthocarpum, which is cancer chemotherapy, and homotoxin mepoxacinate is homoarenginate derivative, which is used in the protein translation inhibitor, especially in the cancer. Mesoprotal Laria tridenta, a cancer chemotherapy. Ingenol 3 angulate, which is Euphoria populus, which cause the activation of protein C kinase and potential application in the chemotherapeutic cancer. Capsaicin, which is a derivative of capsicum annuum. Now it is very much used for the pain management. So likewise, you find number of examples in the market, and there are continue to grow the examples. You can see curcuma longa, which is having about 26 trials for the various cancers. You can see triptolide. You can see the phenoxidil, so which is a synthetic analog of diadesin isoflavin. So we see number of examples. So apart from the drug and drug leads, the phytopharmaceuticals also plays a role in the nutraceuticals. They are the promising sectors with enormous growth. And now if you see the market value for USA, it is about $10 to $37 billion. And Japan is $10 to $14 billion. And you will be surprised to know that just in my biodata, you was trying to talk to me that, I visited Japan. I was visited and I stayed there more than a couple of weeks. I found that very interesting people. And there are a lot of plant-based drugs will be used, plant-based things will be used in Japan. And the Japan population is not more than the Andhra Pradesh population or Andhra Pradesh area. But you see the current valuation of the market is about $10 to $14 billion in the nutraceuticals. Now, nutraceuticals are contributing a lot. So they have a lot of potential. Now I've given United States, Canada, Japan, Europe, Asia Pacific, Latin America, Middle East. Now exactly the global potential. Now what exactly you will be doing? Where exactly you can play a role? So this is what you have to understand. If you try to put it as an extract from the plant, so what are the critical factors I have given? For example, plant material. Can you play any of the role here? You can develop a process, and in the process, the critical parameters like moisture content, degree of processing, origin of the plant material, size of the plant material, nature of the plant material. The solvent, for example, you can play around the solvent, which kind of solvent, the nature of the solvent, concentration, polarity, and manufacturing equipment, manufacturing method. So these things you can take as a novelty and you can produce. For example, if you try to see the solvent, so if you try to see the supercritical extraction, probably you will be able to separate very interesting compounds. Like for example, azadirachtin from the Azadirachta indica, which is used as a pesticide. Now, extracts, simply I don't make extracts. What are the number of extracts available? I have given a big list of plants which are used as an extract. For example, Vidania somanifera. Now I am showing here Vidania somanifera, which is ashwagandha. Now this ashwagandha has got a lot of popularity. Basically, during the COVID time, everybody want to increase the immunity, and now you see and go and see the marketed formulations. There are more than 15 to 20 companies will be using Vidania somanifera extract. And it could be in the form of Chyawanprash, it could be form of immunomodulator. So that's the point of I am trying to derive here is you can see number of plants. You can have very interesting extracts. For example, you see the Humulus lupulus, which is nothing but a hope plant. And now this extract is added into the beer. The beer will be having a bitter in taste. The bitter taste compound is nothing but this. So this is the only source which is added. So you can play around capsicum and indicum, Boswellia serrata, Andrographis paniculata, Allium sativum. Likewise, you take any class of compound and what will be I can use. For example, Tinospora cardifolia, which is called a Tippatiga. The Tinospora cardifolia, Vidania somanifera, as well as Andrographis paniculata are the major compounds of the plants used against the COVID infection. So what are I'm not trying to give you a rosy picture, but there are few problems which are there, which are quality, safety, and efficacy issues. Now, quality is a wholesome factor of statutory requirement, technical experience, and market expectation. If you try to go around these three, the quality automatically comes. When a quality comes, the quality parameters which will definitely make you to have your product superior over the other things. For example, if you want to make a role, probably you can make a role on the technical expertise like quality standardization, quality production, quality testing, quality assurance, quality monitoring. Or market expectation, you can play around what will be the packing standards, what will be the fate of the product. But however, statutory requirements are done by the various pharmacopoeial committee as well as regulatory requirements. Now when I look at a drug, we try to say the safety is more important. Now many of the people think that the plant drugs are very safe, but it's not so, because it's a critical component of quality management. It could be because of adulteration, poor quality, innate toxicity, interaction with other things, misconception, and all are natural safe, incorrect dosing, mistaken use of wrong species. All these problems should be addressed by the pharmacognosy people. Not only that, nowadays the Western market is picking up for the herbal products. So there are simultaneous use of allopathic as well as Ayurvedic formulations are going to cause some kind of interactions, especially cytochrome P450. For example, ginseng and warfarin. Warfarin is an anticoagulant. Ginseng is used as a nutraceutical. For example, if you see the Revitol is one of the product, Hypericum furfuratum, and it's used as an excellent antidepressant. And there are few herb drug interactions, and that's how we have a if you run a PharmD program, everywhere pharmacovigilance is started to increase the credibility of the allopathic as well as Ayurvedic drugs. So pharmacovigilance of herbal drugs should be used for detection, assessment, and understanding, prevention, and understand what are the adverse effects. So the pharmacovigilance of herbal drugs also started. Now herbal drug standardization. Now, what is a standardization? Why we have to go for a standardization is because of the inconsistency in the herbal drugs. Could be regional and linguistic nomenclature, ecotypic, genotypic, chemotypic variations. I just want to give an example here. Vidania somnifera, Rawalpindi serpentina, you will also grow in our area called as Andhra Pradesh, especially in Warangal. It grows in a place called as Pakal. If you take this particular plant from there and try to look for the reserpine, you will not find it. But the same Rawalpindi serpentina from Meghalaya region is having the reserpine and other alkaloids are intact. So these could be chemotypic variation or the other environmental conditions. Now, standardization can be achieved by authentication of proper material. That is one of the major important. Foreign material, organoleptic evaluation, microscopic and macroscopic, volatile matter content, ash value, extractive values, chromatographic profile, marker compounds. Especially if you can't do anything, if you just try to develop a marker compound for a particular plant and try to see that how best you have a chromatographic profile, and this profiling can be done for the newer material which is used in the companies. Pesticide residue, heavy metals, and microbial content. Especially the pesticide residue as well as heavy metal content. These two things are very, very important for the rejection of many of the material which is exported from India. So you need to test before exporting the natural material as well as your material used for the processing. So these are all the general profile I have given. The botanical characteristics like definition, characters, identification, microscopy and macroscopy, phytosophical characterization, identification, tests and assay. And the other side is geographical and biological variation, stability of constraints, all these things, if you can monitor, and then the herbal drugs can be achieved. Now, this is what exactly will be given in your any of the herbal pharmacopoeia. All headings I have given for herbal drug preparations, and this can be achieved if you can try to do the things. So these are the constraints for the herbal drugs. So you need to address them in the quality control, especially lack of trained manpower. I'm sure if I give you the Bixa Virulina, if you come up with a standard monograph by any college teacher, probably you'll say that I am not having enough experience. But unless you do that, you may not be able to get. And similarly, pharmacovigilance data, no certification of raw material, though few of the medicinal plants are cultivated, but we don't have any certification authority. And if you can develop that, probably all these constraints can be removed. Now, just to show you where exactly you can see some of the monographs like herbal drugs. If you see, there are about 133 monographs. I'm talking about European Pharmacopoeia just to see. Overall, today, we have 249 monographs. And if you see constantly from 2002 to date, now 96 has become 249, which means 300% growth is there. So almost the growth is will be grown as 10%. And if similarly, you can see the British Pharmacopoeia, which has now 233 monographs. Now, similarly, USP is also having about herbal drugs 39, herbal preparation 73, herbal medicines 25, national formulary is having about 53, and Chinese Pharmacopoeia is doing wonderful things. And if you see the volume 1 is about 992 monographs. Now, every year they are trying to add new admissions as well as revisions. Now, similarly, Indian Pharmacopoeia currently in 2018 is a pharmacopoeia which has about 129 herbal monographs. So which speaks a volume. If you see it was only 83. Now 83 has grown into the 129, which speaks that it is slowly picking up in India also because of various reasons. Now Ayurvedic pharmacopoeias are also available. If you address your medicinal herbal product as for Ayurvedic thing, now Ayurvedic pharmacopoeia, volume 1, volume 2, volume 3. So you see, there are a lot of things. And similarly, ICMR, Indian Council for Medical Research, also started initiatives for developing standards, and especially they are preparing exhaustive SOPs, standard operating procedures, and you can see the monographs in volume 132 and so on. And they are especially concentrating on filariasis, malaria, diabetes, immunomodulation, hepatoprotection, anti-inflammatory. By this, you can understand what is the current, the preferred area, thrust areas are these. So if you can tap any of the thrust area, the possibility of funding will be more. Now I'm just showing you the volume 1, volume 2, volume 3. You can take about that. Now coming back into the drug discovery from the plants. Now if you see, there is about 61% of new chemical entities comes from the natural products. So which speaks a volume, which means majority of the natural new chemical entities, which is supposed to become APIs, and they are coming from natural products. Now I'm just giving you some examples. Now see atropine. Now classically this atropine has been discovered very, very long back from Atropa belladonna. Now this atropine is used as a starting material to convert into tritropium bromide, which is used in the COPD. Now caffeine, camphor. Camphor is a rubefacient, and probably you laugh at me if you see your Amrutanjan or Jandu balm, tiger balm, all of them will have a camphor. Now cocaine, which has given rise a class of local anesthetics, colchicine, digoxin, ephedrine,
isomine. Excuse me, sir.
Yeah. Yes.
And the screen is not visible.
I don't know screen. I have the same screen I am using. No. Screen visible, kada. I don't know. Now it is. Now it is visible. Is it visible? Gopinath?
No, sir. There are no screens, sir. Yes, yes. Now it's visible, sir.
Okay.
Go ahead.
Okay. Now there are a lot of herbs so as a source of phytopharmaceuticals, and just giving you a reference of 1989. You please go back and check. Now, currently used plants, I'm trying to give an example so that you will be stimulated. Dioscorea deltoida is one yam. I have shown you few pictures of these, and these are still, we use all corticosteroids like hydrocortisone, cortisone, which are used as dexamethasone. All class of steroids are derived from diazogenin, which is a starting material. Now you can see how much of consumption of these things. So you can cultivate yams or develop an extraction method so that you will contribute something. Now I am showing you digitalis purpurea. Even today, digoxin is the only alkaloid glycoside, which is used for the cardiac arrhythmias. The dose is of something like 1 mg for 3 days. So you can see there is no substitute for this. Now, if you can develop any of the plant or any source, you will be contributing. This is a wonderful plant, which is Papaver somnifera. Everybody knows. And we read pharmacognosy of Papaver somniferum, which is an excellent painkiller. And it has under restricted cultivation in the UP and in MP, but it gives a lot of molecules like codeine sulfate, which is used as anti-cough suppressant, and thebaine, papaverin, and you can see other painkillers like similar effects like Isomas niger. It's used as an analgesic. And I'm just giving you an example like Rauwolfia serpentina. Still we use reserpine as antihypertensive agent. Catharanthus roses, there is people who are trying to synthesize vincristine and vinblastine, or can we produce this vinblastine and vincristine in cell cultures? I know how many people are working on that. J.V. Shanks of University of Rice in Worcester is also trying to currently work on tissue cultures. We in our lab at Kakti University also worked. We could not produce this vinblastine and vincristine. However, we can produce other alkaloids. So currently I've just shown you the chemical structure should stimulate the chemist. And I'm sure if you want to synthesize these molecules, which will have 30-40 steps, which could not be synthesized the commercially viable method. Now, Phodophyllum peltatum, a mayapple, which is grows in the Himalayan range, which is used as an anti-cancer agent, especially the skin cancer. Now, this is what Podophyllum toxin. Now, see how many stereochemical centers are there, which complicates the synthetic chemist to synthesize these particular molecules. Now, simple molecule Colchicum antimonale, which is colchicine, which is used in the pain management, especially the gout, as well as it is also used as a DNA methylating agent. Now, why this I'm talking about this plant? The pharmacognosys should excite. Is there any substitute for this? Now, I can show you one substitute. If you try to Google it and try to search Gloriosa superba, which grows very widely, this is also having colchicine. So we need to substitute and develop a newer kind of plants. So this is how it looks like the plant. Now, I give an example of Taxus brevifolia, which has given a wonderful molecule, which is discovered by the MC Vani. The two molecules, one is paclitaxel, as well as the other one is camptothecane. After screening several compounds by the Research Triangle Park of US, and this Taxus brevifolia, I will show you the structure also, and you will understand how complicated it is. And now it is the only drug of choice for the management of breast cancer, kidney disease, and other cancers. And this has given a lead molecule for the generation of newer drugs like docetaxel and so on. Even camptothecane, topotecan, irinotecan has given rise, and these are also very much useful. Now look at, are you able to see the taxol structure? The chemist features, you please look at this structure. I'm 100% sure you may not be able to synthesize this compound that easily, but however, a group in the French called Potier group I synthesized with 96 steps and with the final yield of the compound 0.001%. So which is a challenging for a medicinal chemist, whether you call it a combinatorial chemist or computational chemist or whatever it is. This taxol has a structure. Now, if you see the side chain, here if your side chain is removed, it is called as baccatin. And this baccatin is distributed over more than 10 or 12 plants called as Taxus baccata, Taxus myriami, Taxus chinensis, Taxus wallachiana. So all these plants will contain baccatin, and now this baccatin is semi-synthetically converted by Bristol Myers Squibb, as well as they also have a tissue culture plant to produce these two plants. Now, Camptotheca acuminata, is having a camptothecin, and this also relatively very, very complex molecule. And this camptothecin is distributed among the 4 plants. One plant is called as Campotheca acuminata, which grows in Taiwan, and Nothopodes Pyoetida, which grows in the Nilgiris as well as Pune Sahyadri Hills, Ophiorasa mungos, Ervetemia heniana. So these 4 plants. But however, our research indicates that we tried to work on all the plants. We could not able to produce the camptothecin derivatives in the Ervetemia heniana. However, we produced camptothecin in the Nothopodes Pyoetida as well as Ophiorasa mungos. Now, Lapacho is another simplest plant which I am trying to show you. Now, look at this, a simple, beautiful plant. And this will give you a dye called as naphthoquinone lapachol, which is also a quinone, naphthoquinone derivative, and which can be used in several ways as a coloring agent or a pharmaceutical agent or wherever you want. Now, what could be the approach to discover a drug? So the drug discovery can be done by the identification of the plants, traditional, non-traditional, Ayurvedic system, bioassay guided fractionation, isolation, screening, evaluation, doability, drugability, patentability. That is what you have to do. What are the unmet needs? Even currently, we have about 3 lakh plant species are available, whole universe. Only 30% of them are investigated the plants. Now look at how much there is. Another 2 lakh plant species are available. So you can tap any of them, thoroughly investigate so that you can become a new drug molecule. And if you try to see what are the current number of diseases, which are about 2,000 registered acute and chronic diseases, only 30% of them are curable. Now you have a non-curable diseases as well as you have the plants are available. Only thing is you need to understand the logics and try to work on that. Now, how do you select the plant? The plant selection can be done locally, random ways, taxonomic wise, phytochemical wise, information wise, ethnomedical, serendipity, or documented traditional evidence. Now, plant collection, you should be able to collect the plant in such a way that it will be non-destructive process. But there are few things which are destructive process also. So what you have to do is, in the destructive process, you should see that the plant will have the next generation. You should propagate that, then you should extract. That is what I am trying to derive a point here. Now, leads from the medicinal plants. There are a lot of leads have originated from traditional knowledge-based plant selection to increase the probability of success. So this is given rise several molecules, which I just give you a few examples. Now see, Kelin. Kelin is a compound which is obtained from Ami Visnaga and Ami Majus. Now this Kelin has given rise semi-synthetically modified sodium crimogallate as well as amidorone, which are both of them are having the therapeutic utility. Now, just now I was talking about papaverine. Papaverine is an alkaloid which is present in the papaver somniferum skeletal muscle relaxant. Now you see this has been completely modified and get into the verapamil. Now verapamil is a calcium channel antagonist, and it will be used in the management of hypertension. Now similarly, the another very interesting molecule is galangine, which is obtained from Galangina plant, and this plant is very much used by the traditional people and tribal people, so that whenever their blood sugar goes up, they used to do the juice drinking. And this molecule has been galangine as isolated from the plant, and metformin has become a blockbuster molecule. Now everybody try to use metformin for the management of their sugar levels. Now, having known about that, what are the future direction? What kind of targeted based assays can be performed? Now, I'm just giving you here some examples, and any of the example you can take and try to do the activity and try to take a lead. For example, melanin inhibitory activity. You can do this rat melona cell lines, and which can be, you can make a cosmeceutical products like face creams and skin whitening compounds, osteoporotic activity, anti-obestic activity, hepatoprotective activity, antioxidant activity, memory enhancer like Alzheimer's disease. So anti-diabetic glucose uptake assay. So you can concentrate on one particular assay and you can one particular target, so that you will achieve to some extent in the area of discovery, as well as coming out with some herbal formulation. Now, for example, where I can play a role is just you can play a role, screen the crude extracts, identify for picking the hits, or you can prepare a semi-purified fractions for isolation. You can isolate, structure elucidation, a structure, and then go into the any of the. So any of the area you choose, and you can combine with the other people who are having that expertise, and you can do the research. So this is what everybody knows. You need to identify the lead, then you have to optimize lead development and clinical trials. Now, why I have given this particular slide for you people, especially for the teachers. Now, there are a lot of pharmaceutics teachers are be present. Now, as now the software industry has been revolutionarily changing. Now you can do a modeling, and you can come out with this database with the pharmacokinetic like ADME parameters. Based on the ADME only, there is a company in the Bristol Myers Squibb in the Bangalore, and you can also see the pharmacometrics, or you can predict some predictive analysis using this targeted based analysis or drug delivery. Any of the areas you can play your role, and so that you will be well contributing in the area of drug discovery. Now, having known about this, what are the 21st century priorities? Now, 21st century priorities are protozoal infections like malaria is still a problem. I will show you some plants: filariasis, leishmaniasis, amoebiasis. And I am just trying to give you these diseases basically because these are the diseases where there are no allopathic medication is available which can completely eliminate, like dengue fever or herpes, AIDS, COVID-19, metabolic disorders like arthritis, disease of unknown etiology like cancer, muscular dystrophy, Parkinsonism, any of these self-inflicted diseases like depression. Now we are very ambitious that if you don't achieve that, we'll be depressed. So many of this current generation is undergoing depression. Very frequently say that, I have a depression. But when we are growing up, there was no depression because of the mobile and software. You are not forgotten about your touch with the human beings. That is what it is creating: depression, drug dependency, obesity. There is no physical exercise. That's how it is causing. Now, if you see, these are all the plants which can be investigated, and some examples I am giving, like quinine is still obtained from Cinchona ledgeriana, Cinchona pubescens, Cinchona saxirubra, Cinchona calisaya. All these Cinchona will grow commercially in the Mungpu area of West Bengal. So the malaria is a problem of third world country like India, China, and Africa. Now, which can get like Berberis, Tichlora, Tridenda, and Artemisia anna. So there may be number of examples which I have given you. There may be few compounds, like for example, if you want to go for leishmaniasis, Piagum armala, and Berberis aristata, Teprosia purpurea. Teprosia purpurea, everybody I know, I believe. And the only synthetic drug available is amphotericin B. Amphotericin B is having bioavailability problems, and that's the reason why liposomal formulations are being tried. But the berberin harmalin is also having leishmaniasis activity. Filariasis, only etrasan is available, the drug of choice, but sterbuloside, asparoside will also work, and these drugs are obtained from Sterbula aspirensis. Simple amoebiasis, the drug of choice is metronidazole, but we can also use Syphilis epicocuna, which is also grows along with Cinchona calisaya in the government emitin factory of Mungpu of West Bengal, Colorina antidysentrica, Allium sativa, garlic, and many of the village friends agree me that whenever you have amoebiasis, they will be using the garlic karam. So very interesting examples are there. Only thing is we need to look for the look for the plants so that you will be able to do that. I have shown you the Holarina antidysenterica plant, also very widely grows even in Andhra Pradesh area. Now coming to some of the antiviral plants, there are several plants which are having antiviral in nature. Sambucus nigra flowers, Hypericum perforatum, which I have shown you, Hypericum perforatum, which contain hyperforin, which is an excellent antiviral properties. And this is also having another plant called as Hypericin, which is a novel antidepressant. I have an opportunity of working on this particular plant. Also, this grows very widely in the Himalayan range. Saponeura officinalis, Pavetta ovarians. Now, if you see the avian influenza, people were talking about bird flu virus. Now, I am given here a simple molecule like shikimic acid. You look at shikimic acid, and this shikimic acid is very widely distributed among the plants. Now, the companies have come out with a plant formulation called as Ostelmaver, which is Tamiflu with a 12 steps of synthesis of shikimic acid will give you this plant. And the shikimic acid, very high content of shikimic acid containing plants are there. Illicium vera is Chinese plant. Illicium anisacitum, Japanese plant. Illicium grifti and Illicium simoni is Arunachal Pradesh. And this is an opportunity for India to face a world challenge. And you will be surprised that, couple of companies in Hyderabad, Natco Pharma is one company which produces Ostelmaver also starting from the shikimic acid. Now coming to the AIDS, HIV. Now you see there are few plants which have an excellent properties. Like one of them is, I have shown you, Trichosanthus kirlovi, which is a Cucurbitaceae family. Almost many of the plants which I have shown you, Trichosanthus anguana, Trichosanthus braccatea, Trichosanthus cordata, Trichosanthus curmivaria, Trichosanthus dioica. All these are having a compound called as trichoxanthin, which is a ribose inactivating protein and which shown anti-HIV properties. But the only challenge is that is highly soluble in water. Now, making a highly soluble compound into a formulation is a big challenge, and this challenge can be taken by the people like pharmacy friends or anybody who is interested. And we also have Tinosporia cardifolia, I have shown you, which is called as Tipatiga. Now this is having an anti-HIV activity, Picoraza curro. Now if you see and go and see any of the Ayurvedic formulations, invariably they add Picoraza curro, which is known as Kutki, as well as the Tinosporia cardifolia. Now I'm just trying to show you some immunomodulators. Now what are we trying to say? That the immune system should be good in the case of COVID fighting also. And the immunomodulators can have the plants like Asparagus racemosus, Ocimum sanctum, Bohorevia diffusa, Picoraza curro, and Sismal piliferias, Crinium latifolium, Desmodium gangetum, Vidania somenifera, Tinospora cardifolia, Saida cardifolia, Pureria tuberosa. All these are Rasayana plants of Ayurveda. I've just given you, this is not my knowledge, these are the plants mentioned in the Ayurveda. Now, anti-inflammatory plants, I've given the list of anti-inflammatory plants as well as what are the phytochemicals which can be done. Azadirachta indica, the people say that Azadirachta indica will be having this and that. But specifically when Nimbidin is implicated for anti-inflammatory properties, can I develop a method so that the neem can be only gives you Nimbidin? Boswellia serrata, only boswellic acid, which is an excellent formulation. I'm sure everybody knows that for the management of pain. Vitex negudo and Commiphora longa for curcumin. So all these are having the excellent anti-inflammatory drugs. Now, not only that, the anti-asthmatic plants like Albizia lebec. I don't know how many of I have seen Kakti University. The Kakti University inside campus, if you come, the left and right side, the Albizia lebec plants are there. It will be Leguminosae plant, and you have lot of seeds. Now, ephedrine is used as asthma, as well as this ephedrine is Ephedra geridiana, which is now banned drug, which is basically it is used as anti-obesity. Now there is one formulation which contains amivisnaga, which is a trade name as Intol, We don't know what is a phytochemical for that, but it's an excellent use for anti-asthmatic agent. Now, hypercholesteremics. Now, I'll give you a small example of the case study, like, how the statins have developed. Now, if you try to see the China, the people, what they do traditionally is a red yeast rice prepared in fermentation and washed and cooked non-glutinous rice using the red wine mash, natural juice of Polygonum grass and alum water. They ferment and then they use this. And this particular thing will be containing red yeast rice is nothing but Monococcus pupericans, which contain colectin 3. The colectin 3 is an inhibitor of HMG coenzyme reductase. And based on that, the people have started preparing the other things. Now, like simvastatin, atorvastatin, pravastatin, rosuvastatin, all these have come basically using colectin. Now, colectin, why I am giving an example is you have to understand the traditional people and the people who are there in this particular area, they are not obese. So that is what point I am trying to derive. Now, Ayurvedic traditional knowledge is there for India. Now, Lecinias, which contains Cyprus rotundus, Sarasa lapa, Curcuma longa, Berberis aristata, Pongamia pinnata, Ecorus calamus, Plumbago gelanica, Picorus curru. So all these are the plants which are there in the Lecinias. And now, for example, if you try to see the traditional medicine from modern times to anti-diabetic plants, I have shown you the metformin. Apart from that, the NAPLARET database, if you see, there are 148 potential anti-diabetic plants which are distributed among the 50 families. I'm just trying to give a few examples like Mamodia charantia, Pterocarpus marsupium. Pterocarpus marsupium is nothing but Maddi, Gymnema sylvestra, Sphaerecia chirata, Murea conangi, Salacia pranadis, Sigigum kumi. All these are very wide and significant plants for the management of sugar levels. Now, that is what exactly you have to play a role, you have to find out. Now, the another interesting area is because the diabetes is growing, the constipation is a big problem. There are a lot of export of the purgative plants from India, like Planta govona, because of swelling factor, Cassia angustifolia, Senna, everybody knows, Rohan astrale, Terminalia cebula. So these are all the quality purgatives. Now, if you try to see the traditional knowledge, what a new discovery has given. Now, I'm sure any of you, if you try to talk or try to learn the Ayurveda, there are two formulations which are very widely used, Tripala and Trikatu. Tripala is nothing but Trichopas Thanikaya, Usirikaya, Karkaya. That is nothing but Terminalia bellerica, Terminalia cebula, and Embolica officinalis. Now, this Tripala will rejuvenate the body, entire body. Now, Trikatu is Piper longum, Piper nigrum, and Gingiber officinalis. Now, these three are increasing the bioavailability enhancer. Now, you can see a lot of marketed formulations with this Trikatu. Now, the old drugs are formulated into different formulations because of their insolubility in water. For example, Silybum marianum, now converted into phytosomes, Silybum pas, Ginkgo select, Green select, Centella asiatic, Leucoselect. All these are commercially available formulations, which means you can think about making a drug delivery problem also. Now, if you try to see the anti-cancer plant drugs, now taxus, I have given example, cephalotoxin, Campotheca acuminata, Heliotropum indicum, Catharanthus roses, Podophyllum, MOD. All these are contributed very significantly for developing newer drugs, or they themselves will be having an activity. Now, if you try to look at the perfumes and fragrances, rose oil, lavender oil, jasmine oil, patchouli oil. Now, patchouli is even commercially grown in many parts of the country, and the Indian Institute of Traditional Knowledge is coming out with what are the protocols for the SOPs for the growth and extraction. Now, these essential oils are used as a disinfectants and deodorants. Like they contain pine oil or I am given an example of creosote, which is nothing but the phenols obtained from wood tar for the destructive distillation of wood tree of Fagus grandifolia. Gaikwal and cressol will be there. And you can see the OTC products, Devitt tooth egg drops, expectorant like cremosulpine, creoterpene. So these are a few commercially available examples I am trying to give you. Now, aromatic spices make a domestic kitchen. I am just trying to show you. Now look at, I am not showing your kitchen, but these are the wonderful things. All these things are used as a flavors, which makes your carminatives, especially acting on central nervous system as well as gastric GIT. Now these can be pepper, ginger, and cumin, cinnamon, asafoetida, which contain coriander oil or star anise oil, curcuma oil, or caraway oil or cardamom oil. Now this is what I have shown you before also. Anti-eczematic. Now I'm showing you some classical example like juniper tar, which gives you cadin, which contain OTC drug polytar, pine tar, packer pine tree or polytar. So you just understand that can we make a plant-based product into a OTC product? Now, if you see, I'm giving you a classical example of tiger balm, which is obtained from Gaultheria procumbens, which contain a gaulethrin, which gives you methyl salicylate. And everybody knows that methyl salicylate itself has given rise to many things like aspirin. It's a pain reliever. You have a OTC products like tiger balm, Jntua balm, all these will be containing of gaulethrin. Now, can we use some counter irritants? Now, if you see the Vicks Vapor Rub, I'm sure everybody uses that. It contains nothing but cedar leaf oil, Thuja accidentalis, wormwood oil, absorpin. It's another counter OTC product counter irritant. Essential oils can also play as a diuretic role, juniper oil, and OTC product Vodrinil, which is nothing but bochu leaf oil. Now, diuretics, OTC product for menstrual problems, Fluidex. This is having especially a unique properties which can be used in the menstrual problems. Now, I am showing you painless problem. For all school-going children of those who are parents, they see that this is a big problem. Now you see there are few oils which can have excellent activity like tea tree oil, lavender oil, lemon oil. We also done one of my master student has done some project work to make anti-lace formulations also. So you can make simple formulation, find out, and you can get into that. So this is what exactly I am just trying to derive. Now sedatives have a lot of significant lavender oil, jasmine oil, valerian oil. So we have now, because of depression and obesity and the lifestyle disorders, we are not getting a sleep, especially the software people. So we need to understand that the mosquitoes repellent, you can use a repellent, anything, catnip oil, excellent repellent, anti-parasitic essential oils. Especially I am giving an example like Trichomonas vaginalis, which is one of the biggest problem for women. So they got the fish pathogens, eczema, inflata, and this oil can eliminate all the 3 parasites. There is a reference which also I have given you, 2006 reference. Now, tree oil multifactorial therapeutic uses. Some of them will be useful not only for one particular problem like antimicrobial, expectorant, anti-inflammatory, antifungal, and tonsillitis. All these can be used by the tea tree oil. Tea tree oil is obtained from Malaysia, Lucadendrus or Malica ultrifolia. It is a Chinese horseshoe nut. Now, people think that the pharmacognosy, they don't understand how the pharmacognosis can make a breakthrough. Now, if you see the 2015, Yu Tu, Chinese pharmacologist, has got the Nobel Prize for discovering the artemisinin. Now, you can play a role in the natural excipients. These are all the natural excipients I have given you in the things. You can make into the herbal cosmetics. I've given you the herbal cosmetics, especially skincare. As you can see, you can play a role in the shampoos, anti-dandruffs, or anti-air fall, all those things. I have given Adathoda Vasaka, Allium sativa, aloe vera, Andrographis muricana, Aja directa indica, all these plants, probably you see number of plants. So the list never endings, like air care, like Acacia conicica, Chicakai, Arnica montana, Betula pendula. The list goes on. You can play a role in the natural colors, natural dyes, vegetable origin. Now you can see the anthraquinones, like any of the indigo plant, blue, pink, flavones, carotenoids, like I've given a Bixa orina example. Commercial cultivation is possible now. Carrots, curcumin itself, chlorophyll containing plants, anti-obesity. You can see the number of plants I'm showing you, the anti-obesity. Finally, if you want to become a good discovery man, the nature is the best teacher. Now, I'll just give you an example of zoopharmacognosy. If you try to look at the animals, how they will be treating their own diseases. If you try to look at the human beings, as and when you suffer, you try to go to the medical doctor and address your problems. But cattle in South America will graze in particularly Cestrum diurnum, which will eliminate vitamin D3 deficiency. Similarly, the elephants of Amazon forest, thiarbins, they whenever have an anti-protozoal infection, they go to a tree and they eat 6 or 7 leaves of that plant, and the plants have been isolated with thiarbins. Similarly, if you see the traditional foods of China, uparazin has been discovered from Uparaja serrata. They make a drink, and when they drink, they don't have Alzheimer's disease, and that is nothing but myasthenia gravis. And if you try to look at the tribal people, they use a particular plant, and they use as a Conodrondum tomentosum. That is an arrow poison. So that has given rise to D-tubacorarin. I'm sure everybody knows that skill people use that as skeletal muscle relaxant. Now, Vinca story, if you try to look at, the tribal people use Vinca roots for hypoglycemic agent. But Eli Lilly Corporation of Canada started looking for the molecule, but they could not get hypoglycemic molecule, but they got the few fantastic molecule like vincristin and vinblastine. Similarly, in Indian context, TBGRI, Tropical Botanical Garden Research Institute of Pachapalode of Trivandrum, where Pushpangadan team has observed the people, and the tribal people will be on the other side of the mountain, and they used to climb the mountain, come down for their groceries. While they are going, they exhausted means they take a twig and chew the twig, they get the instantaneous energy. And afterwards, they found out that the Trichopsis jalanicum will be having a similar properties like Panax ginseng. Now, this is nothing but Indian ginseng. Now, only thing is, what is the thing which I want to give a message is, when you look at the stories, the old crow, when she want to drink the water from the pot, is to pelt the stones. Then the water will come up and then drink. In the modern cow, if you see, it will put a straw and drinking. The logic mantra is keep upgrading your skills. With this mantra, I would like to thank Dr. Chekka Gopinath and his team. Thank you very much for giving me this opportunity to talk to the FDP people.
Sir, thank you very much, sir. Excellent talk about the pharmacognosy, especially the Jew pharmacognosy, and the nature itself is the best teacher.
Yes, sir. Yes.
And that is identifying all the medicinal compounds by the observing the animals. No, you gave the narration of the excellent narration, sir. Entire pharmacognosy is covered all the areas.
I don't know. I just want to motivate the teachers.
A teacher has to assimilate this one. In turn, they have to go on with respect to colleges, and they have to disseminate the knowledge to the students.
Not only that, I wish that they take keen interest in working on any of the areas. It doesn't require a lot of instrumentation, only thinking process. That makes them, and they can easily get collaborated with the other good laboratories so that they come up with something that
Yes.
will be good for our pharmacy fraternity at the current scenario.
Any questions from the co-faculty members or my dear teachers? Any questions? Please respond in the chat box or you are giving the unmute option. Sir. My dear faculty members, please. If there are no questions, we'll give the vote of thanks to the Professor Siddhiviresham sir by Pranati.
Sir, shall I give vote of thanks, sir?
Yes.
Thank you, sir. Thank you very much, Professor Siddhiviresham, sir. You gave such an excellent session to us. After your session, we had clear vision on research pharmacognosy and pharmacovigilance, and also we found out new drugs which obtained from global market phytochemicals which are in clinical trials. We learned how much impact of medicinal plants there is business on our country. We have enlisted some top selling herbal extract in world market, herbs which are sources of phytochemicals, how to extract them, structures of some phytochemicals, structure relationship between herbal drug and synthetic drug. What are herbal drugs used to treat cancer, malaria, viral diseases, AIDS, influenza, depression, inflammation, asthma, and diabetes? Thank you very much, sir. We learned a lot from your session. I think you covered most of the herbal plants and brought before us. You did a very good job by giving interesting and excellent session. Thank you very much, sir.
Thank you, Gopinath. I have done my job.
Thank you, sir. Thank you very much.
No problem. No problem. It's my
best.
pleasure.
Okay, thank you, sir. Best regards, sir.
Okay, no problem.
Okay, thank you, sir. Okay. My dear faculty, next session by 3:30. The speaker is already waiting. Babu Nageswara, Dr.